General title there. We've heard a lot this morning, great conversations, great backdrop. We all know that we've got big problems, because, for species that we care a lot about, like the giant sequoia. And to sort of get past these problems or to deal with them, these challenges, we're really going to need good strategies for the next generation, even for very long-lived species. We've talked about that. This requires as much as we can on-the-ground research, detailed information, of course, as much genetic information as we can get, environmental information as we can get, and ecological information. In a day, we've got to make the decisions, right, of like, what are the seeds, what are the mixes, what are the techniques. And I think, beyond that too, though, we also need to be clear about what are our goals, right, for populations. And in this talk, I think I'm mostly targeting species or populations that are really highly at risk, right? Not just general things, general plants out there that we can do cool studies on and test hypotheses on. But think about at-risk things like the giant sequoia. What are our targets? And I think one of our big targets is that we want to try to preserve uniqueness as much as we can, like strike a balance between uniqueness and adaptive potential, right? And adaptive potential means genetic variation. It means maximizing genetic variation, but also somehow keeping sort of a unique identity to populations we want to preserve, right? So in this talk, I just have some points, I'm a scientist, we're all scientists, I think, right? And so we think about things as hypotheses, right? So points to consider that I'm just going to be, and we've talked about a bunch of this stuff already this morning, but so hypothesis, mixed populations are the norm. Okay, that's a hypothesis that I want us all to consider. It's not that we're mixing populations, mixed is already the norm. Okay. Gene flow is mainly adapted to at-risk populations. Okay. So gene flow is generally a good thing. Striking a balance between population uniqueness and adaptive potential is a goal, right? Or maybe a primary goal, maybe one of the primary goals. And we can afford to experiment even with at-risk populations. Okay, so there's an assumption or a hypothesis. So I'm kind of going to just go through these briefly in my 12-ish minutes. A lot of what I'm talking about today we discussed broadly in this paper that was published last year. And I'm not going into a lot of the things that we talked about in this paper, but in this paper we describe what we think is known about patterns of gene flow, effects of gene flow, and implications of gene flow in present populations from our own perspective. I'll end with a little bit of data on an Australian plant that these Australian collaborators and I worked on, did research on, just to bring home one point. Okay, so the point about populations are already mixed. So this is a figure from a paper published in 2016. It's on this monkey flower, Mimulus lineatus, that is restricted to the Sierra Nevada, California, the west slope of the Sierra. And these are 23 populations that represent a broad section, most of the species range. We did a genetic study using microsatellite markers, basically. This figure just shows that there are significant connections of gene flow across these populations. It's a highly selfing plant. It's like 95% selfing on average, right? But there's still this genetic mixing that's always ongoing, even in contemporary timescales. And isn't this interesting, looking at a table like this? But anyway, right. The whole point of this is that these are estimates of recent gene flow from a Bayesian assignment integration estimator don't worry about the details. What I wanted to highlight are these cells that show the estimates for these different populations of this monkey flower, of what the migration is from within the population each year, or not each year, within the past several generations, right? And look, it's varying, you know, upper 85%, into the 60s. If that's true, if that's a true estimate, it means that, you know, up to 30% or more of immigration is happening in a contemporary timescales, right? Even for a selfing species, right? So the point there is just that we shouldn't be looking at populations as like, oh, look, it's from there, it's an ecotype. Think of populations like cities, right? They're full of migrants. They're full of people who just got there. They're full of people who've been there for generations with their families. That's what populations are, okay? So you gotta get out of this fixed thinking of like, oh, if we mix, well, that bad. They're already mixing, and they've been mixing for millions of years, right? So that's a point. The other point about gene flow being beneficial is when we're talking about at-risk populations, we're mostly talking about small populations, okay? Small populations are the ones that are at highest risk for whatever it is, you know, of risks of going extinct, risk of being extirpated. There's a figure from you know, a Hoffman et al. Paper that basically lists some risks associated with small population size, right? There's a lot of different risks: genetic risk, allele effect risks, you know, ecological risk even. But you know, one listed here is reduced gene flow across the population structure. Okay, so basically, not having the idea here is that not having much gene flow is a risk, right? The other side of that coin is that regular gene flow is a positive thing. And I'm just pointing out some, you know, some empirical studies and some theoretical studies. I mean, we found in a paper gene flow is good to little populations on the edge of a range of a monkey flower. Ben mentioned this this study last night, which showed pretty much the same thing at a northern edge of Clarkia species range, where gene flow from warmer environments that matched the current climate that the plants were growing in was beneficial. There's studies showing that it's not just even about population persistence or vigor, but, you know, cool studies showing that assisted tree migration might actually like improve carbon sinks and like climate change response. So there's other like even much larger emergent properties from beneficial gene flow or gene mixing. This is just an example. Genetic and habitat rescue, improved population viability in self-compatible plants. When you do a literature search, I don't know if it's just, you know, because people like to publish positive results, but there's a lot of positive results out there for gene flow. Another one, endangered species with a mixed mating system, and then a recent meta-analysis about genetic rescue leads to higher fitness as a result of heterozygosity across animal taxa. The last few papers were all from the past year, right? So there's a lot of data starting to come out, and cool modeling studies to show the benefits. This is a figure that I show a lot in talks. I'm really a big fan of this paper and this figure. But, you know, background reading was the Aitken and Woodlock paper, and Christy Brigham mentioned the 20% idea or hypothesis. This is just an idea that, you know, in this cool paper that maybe as a target of management is to basically understand what the background patterns of gene flow are and to try to mimic those, right, in your restoration efforts. And if you think that local is good, great, put local as the biggest proportion in there. Put some mixture of intermediate, but yeah, you might want to have some long distance stuff to add some real novelty, especially if it's from climate regions that we think are going to be beneficial in 10 years, 50 years, 1,000 years for a giant sequoia. I like this because it already incorporates the idea that populations are really a mixture. They're not just like types. They're a mixture. Okay, and then I'm just going to end off with our finally some data, a little bit of data. For my postdoc, I went to Australia. It was amazing. I mostly like tramped around, Australian Alps and got involved with cool projects. But I got to work with this species of trigger plants, the Lydium arborea. Cool genus. It has this like pollen column that like actively smacks a bee, or a pollinator once it lands on it, and sort of establishes pollination that way. And you can like trigger the plant. It's really cool. But we did a study. We were interested in this sort of experimental gene flow across populations of this trigger plant in the Australian Alps. So in this little region of Southeast Australia, where these mountains, like Snowy Mountains area, they're like kind of like the Appalachian Mountains. They're super old. They're about 6,000 feet high, and you have these these cool isolated populations on the tops of these mountains. And so we went to 3 mountain regions, and we did sort of the full factorial experimental gene flow across all populations, within ranges, between ranges, within populations, etc. We had these huge plans, of course. We were going to put them into like open-top chambers, and we were going to put all these stresses on them. Time just got away. It took years and years to do this. So what we finally got out of it was just doing the crosses and growing the offspring and just measuring their responses in the greenhouses. That's it. And that took years. That was after I left the postdoc. They were still growing plants and measuring plants. And the top panel here shows the number of seeds produced within populations, within ranges, and between ranges. And you see the sort of expected potential outbreeding depression, right, in the number of seeds produced. When you look at mean seed mass, it's sort of that outbreeding depression signal goes away. Just basically, there's no real effect. You might see a little dip between and the faraway crosses in mean seed mass. But when you get to the plants that actually got through to growing and surviving in the greenhouse, all the signals went away, and this is plant size, which is a proxy of fitness when you have a perennial plant that's young, right? We're far away from understanding lifetime fitness for this plant, and obviously this is only in a greenhouse. But look at, what I want to point out is we have really high plant size variance maintained with even the FAR gene flow treatment, between mountain ranges, and I think like just even, we get carried away with the means and mean responses. We need to really think about variance too, You might get a little bump by a certain treatment in the mean, but what does a variance look like? And not getting into this in detail, but there's this, long-standing idea of Fisher's fundamental theorem of natural selection, which is that fitness variance basically predicts the adaptive potential. So basically, the higher the variance in fitness among individuals, the higher the potential for future adaptation. If this was all the information we have for these crosses and these different ranges, I would say there's no reason not to try some long-distance gene flow and restoration projects for this plant, especially, if it's really blinking out and we need some, we're desperate for some solutions. Wrapping up. Take-home points. At-risk populations likely suffer from isolation in small sizes, genetic drift, aka not too much gene flow. So that's just a working idea, right? Gene flow patterns are knowable and are likely changing. So even when we go out now and we do estimates of population genetics and we say, oh, these populations are connected this much that way, etc., that could have been altered from 30 years ago. There's cool studies showing that, like California bighorn sheep, their genetic patterns are already altered from roads and building. You're not looking at a historical pattern in gene flow. You're looking at contemporary patterns of gene flow, and you got to think about that. So when you say, anyway, high fitness is great, and we want that, but high fitness variation is essential. for adaptation when you don't know what the future holds for different environmental changes, right? High adaptive potential. And so we end up in this paper saying, mix it up, which reviewers really hated, but we finally got it through. But intelligently, right? Because a lot of people, it's scary. I get it, it's scary. Like, we don't want to mix things, right? But they are mixed. That's the point. They already are mixed. Don't overmix, but understand how mixed they are. Gene flow is natural. It's good for adaptation. And it's pretty much an essential tool. Okay, so acknowledgements and thanks.